With all of our passive processes, remember there were two things that were important to classify them as passive. Number one, it did not require the use of ATP. Now, by now you should know what ATP is, so I'm going to quit defining it. Last time, ATP is human gasoline. It is our energy storing molecule that fuels our action. Okay? The other difference was we were moving things along a concentration gradient from where there's more of something to where there's less. If we were moving solutes, it was diffusion. If we're moving water, it was osmosis. Active processes are the opposite. They do require ATP. Simple way to remember that, if you're active, you're requiring energy. Okay? The other thing is we're moving things against the concentration gradient from where there's less to where there's already more. So we have two types of active transport. They're called primary and secondary active transport. And what determines which it is the source of the ATP. Primary active transport uses energy directly from breaking down ATP. So this breakdown provides a phosphate group after we break that phosphate off, right? And it's going to be added to that membrane transport pump, which causes that pump to change shapes. That allows the movement of the solute across the membrane. So we call that action phosphorylation, which simply means to add a phosphate group. So we're taking the phosphate off ATP and adding it by phosphorylation to the substrate, which is the pump. Okay, by now I bet you can figure out what ion pumps do. Pretty good when the name tells you everything, right? They pump ions across the cell membrane that help maintain the internal concentration of ions in the intracellular fluid. So one example of that is a calcium pump that pumps calcium across the plasma membrane to get out of the erythrocytes. When calcium accumulates inside our red blood cells, that can cause the cells to become very rigid. Now these erythrocytes have to be very flexible because a lot of the little capillary beds that they go through are sort of like Fat Man Squeeze if you've ever been up to Rock City in Tennessee. It's a tiny little passageway that you have to sort of bend and flex yourself to get through. So if erythrocytes lose their flexibility by accumulating too much calcium, then they can't get easily through those small blood vessels. So the ion pumps get rid of that excess calcium for the cells. Within a normal cell, there's more concentration outside of the cell membrane, more concentration of sodium outside the cell membrane, and a greater concentration of potassium inside the cell membrane. This is maintained by a special kind of ion pump called a sodium-potassium pump. It's really an exchange pump because it moves one ion into the cell against its concentration gradient while moving the other ion out of the cell against its concentration gradient. If you walk through the steps in this image, you'll see that 3 sodium ions are pumped out of a cell for every 2 potassium ions that are pumped into the cell. So the cell's got to use ATP to maintain those levels of the ions on each side of the membrane so that we keep more sodium outside and more potassium inside. This pump is also sometimes called sodium-potassium ATPase. What does ACE tell you? You hadn't been studying if you don't know this. ACE tells you it's an enzyme. So remember, these are proteins embedded in the cell membrane. So this protein, this pump, is also an enzyme that splits ATP so that it can power the pump to move that sodium and potassium. There's a 1 to 2 to 3 ratio in the way these pumps work. 1 ATP is required to pump 2 potassium ions into the cell and 3 sodium ions out of the cell to maintain that electrochemical gradient. Are you wondering why that's important? I promise we're going to cover it in great detail later, but I'll tell you now the importance of that. That sodium being in higher concentration outside helps us to maintain something called a resting membrane potential. So we have more positive ions right outside the cell membrane. We have more negative just inside the cell membrane. That allows those cells to utilize that electrochemical gradient to do something called depolarize. Now, that's a big old word, but just to keep it simple, a depolarized muscle cell contracts. A depolarized nerve cell fires a nerve impulse. Pretty important stuff. We're going to get there. With secondary active transport, we're still moving things against the concentration gradient, right? So that means we're still moving from where there's less to where there's more. Well, the sodium-potassium pumps that we previously talked about provide the energy that is needed for this to happen. So it uses the energy from the movement of the second substance down its gradient. So it's moving more than one thing across the membrane. Okay, that's the important thing here. So it's not just moving one substance across the membrane. We're moving two. So the movement of that first substance across the membrane provides the energy for movement of the second one. Okay? All right. So we have two types of transporters that do this. A SymPort moves both of the substances in the same direction. So they both come into the cell or they both go out of the cell. An antiport, anti means against or opposite. One substance is moving in the cell, another is moving out. And again, you have your illustration beside the words there to show you how that works. Vesicular transport's also called bulk transport, but it involves a vesicle. Vesicle simply means bladder, so think of it like a hollow container, right? So the vesicle is a membrane-bound sac filled with materials. I call them sacs all the time in my lecture, okay? So think of a vesicle as just a container, and the container itself is made out of the phospholipid membrane, okay? So there are three major processes that use vesicles. Endocytosis brings things into the cell. Exocytosis, think exit. Endo, come in, exit, go out. Exocytosis, osis means condition, so out, cell, condition, exocytosis. Endo, in, site, cell, osis, condition. Y'all, I love words. Trans means across, so this is a combination of endocytosis and exocytosis that's used to move things from one side of the cell to another, and typically into a cell from one side and out of the cell on the other. Large substances like hormones, neurotransmitters, and waste products, they're pretty big. It's hard for those to get out of the cell unless we package them up. So we package them into a vesicle. The vesicle fuses with the plasma membrane and releases the contents. Now remember, your membrane is made of phospholipids, so the vesicle is made of phospholipids. When a lipid meets a lipid, they merge together, and if one is carrying something, the contents then just sort of fall out because now you've just got one big lipid, not a vesicle and a cell membrane. Now the opposite of that is endocytosis, where we bring things inside the cell by packaging it into a vesicle. So you can see on the right-hand side, we've got this green glob. It sort of hits the cell membrane. The cell membrane forms a pocket that's called invagination, and it closes up, encircles the little green blob, and brings it into the cell. Well, that is endocytosis, bringing something into the cell. We have three types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. Anytime you see phage in anatomy, think eat. So osis, condition, site, cell, phage, eat. So this is when we have the cell membrane engulfs large particles by circling it into a little vesicle and bringing it into the cell. So cellular eating. Now, typically we bring things in, and with phagocytosis, it's going to be digested because your phagocytes are part of your immune cells. And what do? They travel around and find things to eat that shouldn't be there. So they kill microbes or cellular debris. They break it down, okay? So the vesicles contain some chemicals in there called lytic enzymes. Lytic means to lyse, so we call these lysozymes if that's what they're doing. They're phagocytizing the product to bring it in so it can be, quote unquote, digested and eaten. Now, it's really not eating it, but phagocytosis brings it in so it can be digested or broken down. Pinocytosis, sometimes we call it cell drinking, because this is when the cell membrane invaginates, creates little pockets that completely circle and close off, forming a vesicle to bring liquids into the cell. So the liquids will contain the dissolved solutes, and all of that comes into the cell. So we usually form multiples of these vesicles at one time, and most of our body cells do this frequently. So the way to remember it, Pinot, if you drink wine, a Pinot Noir is a wine. What do you do with it? You drink it. Pinocytosis, condition of cell drinking. Receptor-mediated endocytosis uses receptors in the cell membrane to bind to molecules that are in the interstitial fluid and bring those molecules into the cell. This allows the cell to be able to bring in bulk quantities of certain things, even though those substances may not be very concentrated in the interstitial fluid. The transport of cholesterol from the blood to a cell is an example of receptor-mediated endocytosis. So when cholesterol is in the blood, it's attached to a protein called a low-density lipoprotein. We commonly just call them LDLs. LDLs move from the blood into the interstitial fluid, and then they bind the LDL receptors on the cell's plasma membrane. Then those receptors activate the process that causes the cell membrane to encircle the ligand. Okay, so that's going to be our LDLs, our lipoproteins there. The cholesterol will bind to those, and we carry them, then that complex binds to the receptor. That causes the invagination of the cell membrane. It closes off, forms a vesicle, and bam, we just brought that cholesterol into the cell. Familial hypercholesterolemia. Boy, that's a word. Let's look at it and see what it means. Familial means of the family, hyper, above, cholesterol. Anytime you see emia, E-M-I-A, that means condition of the blood. So, too much cholesterol in the blood. This is an inherited genetic disease where the receptors or the proteins of the LDLs are defective. They don't work right. They're not made right. Well, this interferes with the normal bringing that into the cell, that receptor-mediated endocytosis. Now, this results in a lot of cholesterol being in the blood rather than being taken into the cells and used. This can cause a condition called atherosclerosis, as you see here in the image. It's the buildup of that cholesterol fatty plaque within the blood vessels. Now, you can also look at that and see what that does to blood flow. It's going to restrict blood flow to the tissues that are on the other side of that big fatty plaque. This is going to lead to a great increase in the risk of a heart attack. And this last slide is your concept overview that covers all of your active and passive processes. This is in your textbook on page 122 and 123. If you just walk yourself through this, it will give you a nice little summary of these processes.